Good afternoon, everyone. My name is Mark Nardone. I'm director and co-founder of Bio-Trac. Welcome to our Thursday weekly webinar series. We ask that questions be submitted in the Q&A box. They will be addressed at the end of the presentation as time permits. For those who might not be familiar with our program, Bio-Trac offers hands-on training workshops for research scientists. These workshops are team taught by active researchers focusing on the latest relevant techniques that are necessary for laboratory research. We were originally based at the National Institutes of Health for 30 years, and Bio-Trac is now located on the Montgomery College Germantown campus, the Bioscience Education Center. These programs are open to the scientific community. Since the program's inception, we have provided hands-on training to over 17,000 research scientists. Along with our hands-on training workshops, we continue to serve the scientific community through custom design training programs, biopanel networking events, weekly webinar series, symposiums, hosting government and industry retreats, as well as other key scientific events and outreach initiatives, such as the Maryland Science Olympiad. Today, it's my pleasure to introduce to you Dr. Allissa Dillman. Dr. Dillman serves as the training strategist for the Center for Information Technology Cloud Services team in support of the National Institutes of Health STRIDES initiative. Dr. Dillman is focused on enabling the utilization of cloud capabilities by generating training content relevant to the biomedical community and creating such events such as codathons. Dr. Dillman also serves as an outreach coordinator for the Office of Data Science Strategies at the National Institutes of Health. I've had the pleasure of working with Allissa for the past 5 years, as she is a key contributor to our R and next-gen sequencing related workshops. These workshops have provided hands-on training to over 350 research scientists from local, national, and international institutions. Without further ado, welcome Dr. Allissa Dillman, Harnessing the Power of the Tidyverse. Allissa, they're all yours.
Thanks, Mark. All right, so the first part of my presentation will be kind of a brief intro to things, and then the rest of this will actually be a hands-on demo showing you exactly how awesome our studio and the Tidyverse can be, and how it can really help you automate your visualizations and workflows. So the first thing I wanted to talk about was just to briefly mention what R is. So I imagine some of you are joining this talk because you're just kind of interested in, why should I even spend time to learn, maybe this is another language, maybe you already know other languages, programming or coding languages. Or perhaps you're just, used to Excel and you know Excel, and so you want to kind of know why would I do this? So R is a programming language and an open source software environment that can manipulate data, visualize data beautifully, and perform statistics. And actually R kind of originally was created for this statistical community, and has a lot of really strong packages around statistical analysis. The other thing that I really love about R, and what really makes R a wonderful community, is that it's free and relatively easy to run in any environment. And you can download it at the rproject.org. So that's the link there. Oops. All right, so I'm specifically going to be using an add-on called RStudio. So what is RStudio? RStudio is an integrated development environment. Okay, so what does that mean? It's the idea that there's software that has the R language, but it also has things like a graphical viewer, file tracker, code notebook, et cetera, all basically so you have this interactive one-stop shop for all of your R coding needs. And you would download this separately at rstudio.com. And like I said, this really makes for a truly interactive experience, so you don't have to ever leave the RStudio console. All right, so we're specifically going to be using and talking about the Tidyverse. So what is the Tidyverse? So the Tidyverse is a specific collection of packages in R that are really focused on the data science aspects of exploring, visualizing, and manipulating data, and that kind of iterative process that we do in data science all the time. And all of these packages kind of use the same language and use the same interactions, so they really kind of sync well together. Now, Tidyverse is not part of the base R framework, so you do have to install it. So the first time that you do this, so if you want to do this at home later, would be that you would need to use the command install.packages Tidyverse. And then it would be saved in a directory called your library. And then when you want to actually turn on this package set, you would select that by saying that you want to turn on, in your library, turn on the Tidyverse. And the reason for like kind of turning on and off packages in R is, at the end of the day, R is free and open source, so it's not necessarily regulated in any way, shape, or form. So everybody can write their own packages, they can write their own expressions for those packages. But the problem is, if I write an expression called gene express and it does one thing, and then somebody else writes a package for single cell data and they write gene expression does something completely different, these things would clash because R wouldn't know what to do. So being able to turn off and turn on packages can really help kind of make sure that everything stays synced. The other nice thing about a package is it's not just the code used to run the package, it's also you know, usually the data associated and other things like that. Are people not being able to hear me?
No, you're fine, Alyssa. Somebody was
Okay.
having feedback on their specific one. My suggestion is for them to log out and log back in, but everybody else
Okay.
hearing you crystal clear.
Okay, cool. Just checking. Thank you. All right. So as I said, there's actually, this is a collection of 8 different packages, and we're not going to have time in this very short demo to use them all, but we will use a couple of them. So we'll use dplyr. One of the particular commands we're going to use in dplyr is select. So this is actually the command that you would use. You would say select, and then you pick whatever it is your data set is, and then you say which parts of the variables you want to select. This can be specified as either like the numerical column number or as the column names themselves. And here's kind of just a little visual to show you. You start out with the table with all the data, and then you're kind of selecting a subset of it to put into a new table. dplyr also does a whole bunch of other things, but like I said, we're just kind of using this one command. Another package we'll be using as part of the Tidyverse is the tidyr. We're going to be using the gather command. This is set up as gather. Again, you select whatever the data is that you want to utilize this command on. You're going to have a key and a value. So I'm going to walk you through this because this is kind of actually the more complicated part of this whole talk is kind of understanding the structure of data. So this gather command transforms the data from wide format to long format. And what that means is, you know, originally each column name is a key. So each column is a variable, and then each row is a particular observation or patient or whatever it is. That's the wide format. When we transform this into long format, you can now see that all of these keys, which were the column names, are now all in a single column called key, and then their corresponding values are in a single column called value. And then you will see the same observation multiple times for each one of those original variables. And the reason why we're going to use this is being able to manipulate data in different ways can make it easier to automate and graph, to visualize things. And then the final thing we're going to be using is ggplot. This is a really great graphical program. It will make publication-ready graphics. And the command is ggplot. Again, you select your data, and this AES, aesthetic, basically means the variables that you choose, which will map to visual properties. So you'll pick whatever X data you want, whatever Y data you want. You'll pick colors, shapes, sizes. So it's just to map your variables to visual properties. And then ggplot is layered. So then you will add what specific kind of geome type you want. So that's the type of plot. And the reason why this is kind of a twofer command process is because, you know, you may have data and you don't want to change this part of your command, but you may want to look at different types of visualization. So the nice thing about this is this first part of your code stays the same. You only have to change the geome type. So that's why it has this layered process. So you can kind of just add to your graphic rather than having to re-architect your entire code each time. All right, so that takes us to the end of the slideshow portion of things. Now I'm actually going to do the demo. And it takes, so I'm going to be using the RStudio Cloud. So you can actually, you could actually, in theory, access this even from things like an iPad or whatever, because it's just a web-based version of R. I'm using this because, unfortunately, my home computer died, which is what I had R on, and my work computer, I don't currently have access to download because you have to download the software. So the RStudio Cloud can be a really great way for you to work in RStudio without having to download things, or you know, for instance, if you work on a different computer here versus there, this will travel with you no matter where you go. Again, it's all free. It's really great. All right. So now that we're here, I just want to say that, so this is our console where we'll actually do our coding. We have a panel over here which will show you like the history of codes you typed. It'll show you any objects that you have, so data sets, and a couple of things. And then down here, this panel shows you all the packages that you have downloaded, and then if they're on or off. And then, you know, things like it shows you your file structure. It shows you, you know, if you need help, you can query for help here. Plots that you make will show up here. So like I said, this RStudio is really kind of this completely integrated system where you can see everything. So the first thing I'm going to do is I'm going to turn on some libraries. So I'm going to turn on the Tidyverse. Right, so this comes up and it's telling you it's attaching or turning on all of these packages that are part of the Tidyverse library set. The other thing I'm going to do is I'm going to load a package called Survival, because there's a data set there that I'm going to use, which is really useful for kind of exploring clinical data. And that data set is specifically called PBC. So, okay, what in the world is this data set? What is PBC? What's going on here? So anytime you're confused about what happens in R, you can actually just do question mark, and then I'm going to say, what is PBC? And if I do that, you see here this window, the help window pops up, which actually tells you, hey, PBC happens to live in this package called Survival. And it's this data set from Mayo Clinic primary biliary cirrhosis data. You know, that it's, you know, has this many patients, you know, it's been going on for this many years. And then it even tells you information about each column and what the data is in each column. So that's really handy. If I want to actually look at it like I'm used to looking at a table in Excel, I can actually do the command view. Make sure you have a capital V. R is case sensitive. That means if it's lowercase, it will call things in lowercase. So if you tried to use a lowercase v, it wouldn't work because it's case sensitive. So if I type in view, this window pops up, which actually shows me basically a spreadsheet or table of this data right here, right? So you see all of these values. Oops, that's not what I meant to do. Sorry. Here, I'm gonna pull this up a little bit more so you can see the code a bit more. All right, so now we know what data we're working with. The first thing I want to do is just do a simple plot using ggplot. So the first thing I say is that my data is PBC. I want my aesthetic to be, let's say we want to plot age. And all we want to do is look at, you know, what is the age range of these patients? So let's do a density plot. So now we see over in this window here, we now have a plot of the overall age, you know, range of the patients here in this data set. So, you know, it starts off at around 20 somewhere, and the oldest patient's around 80. You got your average age range around here, like 55. So that's nice. We kind of get to see what our overall distribution is for age. The other thing I forgot to mention is you see now here up in the environments section, you can see there's now two data sets, one called PBC, one called PBC-seq. We won't deal with that, but it tells you kind of a summary of that PBC data here, that there's 118 cases and there's 20 different variables. All right, so the nice thing about R as well is I don't have to keep retyping things. If I actually just hit the up and down arrow, it'll scroll through my history of commands. So if I hit the up arrow and go back to this previous command here, and let's say I want to add a variable to my graph, I can just go here, and then I can say, let's say I want to add a color, and I want to look at age based on sex. So now I have a plot which has broken down my data between the age range of males versus the age range of females. So again, this is really nice. It's really iterative, and you can really just go back and tweak one little part of your code to kind of build on your graphics. So then you can really explore data in this very iterative fashion. Another nice thing you can do, like let's say you're like, well, I don't really like them, inside of each other, like both of the lines in the graph. That's really difficult for me to interpret. I want to see them side by side instead. The nice thing about R, again, is you don't actually have to generate each graph separately. Instead, you can use something called facet wrap. So facets basically divide a plot into subplots based on the values of one or more discrete variables. So here, our discrete variable would be sex, so male versus female. So we can use facet wrap to actually pull out two separate graphs for them. So we can take our previous command here, and we can use the facet wrap command, and we can say that we want to wrap around sex. So now, instead of having both of these graphs in the same density plot but colored, we have them pulled out as two separate graphs. Now, this could be really useful if you have like a scatter plot with lots of data or something like that. So this is really handy and kind of generates multiple plots with just a single function. So that's really nice. So let's say I wanted to look at all of the continuous clinical variables in this table. Now, I would have to actually call them one by one because the data is in this wide format right now. But if I transform the data into long format, I could actually use this facet wrap setup to graph all of the continuous variables at once. So that's exactly what I'm going to do. I'm going to generate a new data set called PBC long. And I'm going to say that I want to select my PBC data set. And I'm going to use something here called a pipe. That's this percent greater than percent sign. That's a pipe. And what that means is it will take one command and immediately go to the next command. So rather than writing out a separate table for each thing I'm going to do to this data set, each manipulation, it'll just pipe it into the next thing. So for instance, I want to select column 6 because that's the column for the sex information. So I do want to keep that as my one discrete variable. But then I also want to pull out all of the continuous variables, basically column 11 through 18. So that's the first part of my command, which is just please subset this data. And then the second part of my command will actually be this gather command that I talked to you about, which is taking that data from wide to long format. So once I've selected just the continuous variables in the sex variable, I now want to do that transformation. So I'm going to say that I want the column to be called variable. I want the other column to just be called value because that's easy to remember. But the one thing I want to do is when I'm taking this from wide to long format, I want all of the continuous variables to be transformed, but I want actually the sex variable to stay as its own column. So I'm going to run this command, and we'll actually look at the data so it makes a little more sense when you actually see it. So if we use the view command again, PBC, maybe I shouldn't type so fast, long. This is our new table, right? So we went from this wide format where every column's a variable down to this long format where we do still have sex as its own column, but now every variable name is in a single column, and then the value for that variable is in the next column. So now that the data is in this format, we can actually use that facet wrap command and make a whole set of graphics for all of these different variables. So let's go back to ggplot. We can do PBC long. And then the aesthetic here, so that the variables that we are going to be viewing will be the value, right? So that's the column that actually has the numerical values for these continuous variables. We're still going to do a density plot because all we want to do basically is take a look at all of these different variables and kind of what their overall distributions are. And then the last thing we're going to use is this facet wrap. This time we're going to wrap around that variable column. And then the only other thing I have to add here is scales equals free, because in the original command, basically if I'm trying to compare two graphs side by side, and I really want to actually say, like, is this, you know, is there a different distribution of male versus female? Like, I want them to be on the same scale, so that's actually meaningful. But here, I'm not trying to compare the different sets of variable graphs. Here, I just want to see the free distribution of each graph. So if I set the scale to free, it will automatically pick whatever whatever range is appropriate for that particular. All right. So now you see here that we have, you know, 8 graphs, all made simultaneously by one single command. And we can actually quickly assess the overall distribution of all of these different clinical variables. So this is really handy. The other nice thing is, because we still have that sex variable as its own column, we can do what we did in our original demonstration, which is we can add back the sex as a color variable. So we can actually go back here and we can say color equals sex. And we now have 8 different graphics where we can simultaneously kind of look at, you know, is there any differences in the distribution of these variables in males versus females? And the really cool thing, and I really wanted to strike home with this demonstration about R and RStudio, is really this idea that, you know, if you really think about it, there was basically only 2 commands I needed to do to generate this whole series of plots and to really assess quickly all sorts of information, right? So that was literally just, you know, just this command here and then the ggplot command. So with 2 simple commands, everything is iterative, everything is automated. I can, you know, go back and explore things very, very quickly. So that in a nutshell is really the power of R, and really the power of, I mean, and to be fair, that's true for any coding language. That's not just R, you know, that would be Python and Java and all that fun stuff. But really that's something that's useful here, is like, once you create this, you could even then, you know, take a different data set and you could automate this, because all you'd have to do was swap out the data set, and then you could still, you know, you could make graphs on that and things like that. You can also, you know, you would save all of these commands, and then you could write it into a notebook, which you would do up here. So you can actually add like an R notebook or an R script or R markdown, whichever you prefer. They do slightly different things. But if I just wanted to do like a simple R script, you could actually take all of this. I mean, I would type it in there really, but you could take all of this, copy and paste it here. You would save this file, and then you could, you know, open this, give this file to any one of your collaborators. You could open it up for yourself later, and you can go through and see all of the work that you accomplished. But yeah, that's basically it. That's the end of my little show and tell and demo, and I really hope this gets you excited about, you know, learning more about R and how useful it can be in your work.
Thank you very much, Allissa. Great presentation, and always a pleasure to work with you on these events. We do have a couple questions. Let's see. One is there a way to know what data sets are available in the library? Let's see. We knew PBC data beforehand and loaded in the data. Is there a way to know what data sets are available in a particular library?
Yes, so you could actually either go to the library vignette on, so there's 2 kind of basically large repositories for R packages. So there's CRAN, and there's also the bioconductor repository. So you could actually go to the vignette there for a particular package, and they'll tell you what kinds of data sets are in there. You can also go to the help when you type in, like if you were to type in the name of the package itself, it would tell you, let's see if I can do it here. So it will actually, if you type that into it'll actually pull up the package, and inside this vignette or this walkthrough, it should kind of, if they did the documentation well, it should kind of tell you what is in that package. There's also a bunch of data sets that are just like in R itself. And actually, if I remember, I'll put a link in my slides that has basically like all of the free data sets that are not even like in a particular package in R. They're literally like in R itself, like base R, just all of these different data sets. And funnily enough, I actually used one the other day because I needed to manipulate some state data, and they had already this data set in R that was, had a whole bunch of state information. So there's a bunch of really useful data sets in there. Great question.
Next question, Dr. Kelman noticed under help, learn R online choice. Is that a document, a video, user manual? Is that a good way to learn R?
Yeah, so there's a really awesome set of tutorials for R actually in RStudio, where they have like interactive tutorials. And actually, again, what I can do is, so I believe that Mark will be distributing the slide deck. What I'll do is I'll have a final slide that has all of my code for this demo, but I'll also put links for the answers to your questions on here. So there's I think you can find it pretty easily when you're in our cloud, but I'll put it in there, and it's basically all these little hands-on demos for you to work through while you're in our RStudio cloud. There's also the R for Data Science, which is a free PDF that's really helpful. And again, I can put the link there.
And I'll also interject, we have a two-day hands-on R for research scientists and our workshop designed for research that we offer twice a year at the Bioscience Education Center, and Allissa is one of our key contributors. Last question, is it possible to make long to wide data using Gather?
So that would be, so you would do the inverse of Gather, which I think is, oh gosh, I always forget what there's an inverse of Gather, which is, I think maybe it's spread. There we go. Yes, it's spread. So you would use spread if you're doing long to wide, and Gather if you're doing wide to long.
Very good. Just want to thank everyone for attending today's event. Next week, you'll receive an email from my office receiving further information regarding the recording of today's event, Dr. Dillman's contact information, as well as a PDF of today's presentation and script. Next week's webinar will be given by Dr. Fatah Kashanchi from GMU. Fatah is an expert in exosome research and one of the key contributors for our hands-on exosome workshop. His talk will focus on separating viruses from exosomes, including his current research on how to separate coronavirus from exosomes. I hope to see you at next week's webinar. Enjoy the day and be safe.
Thank you.